Hello everybody, this is Jason here at the Fort Bend Children's Discovery Center, and I'm inside the Ann and John Johnson How's It Work Gallery, and I happen to be sitting at the Science Station, which is sponsored by May W. Tape DDS Pediatric Dentistry and the Gulf Coast Medical Foundation. And I am here today to extract DNA, a wonderful, wonderful polymer, because all this week we are going to be talking about polymers. So, what is a polymer? A polymer is a long strand molecule that has repeating subunits. And DNA happens to be one of the more famous polymers because A, it's really, really long, and B, it has a lot of repeating subunits. And these are repeating subunits that you actually will learn about someday. But I'm just going to give you a little hint of them right now. First of all, where is DNA found? It is found inside each and every single one of your cells in your body. In your brain alone, your nucleus, you have 86 billion neurons. Think about your whole body, how many cells you have, and how many nuclei you have, and how many DNA strands are in each of those nuclei. The reason DNA can fit inside of your nucleus, even though it's really, really long, is because it's in a double helical strand that can tighten up really, really, really tight and become really, really small. If you untighten one of those double helical strands and stretch it out into one single strand, it is 2 meters long. That is almost as tall as I am. Each and every single one of your cells has this DNA inside of it. And inside that DNA are little codes that actually tell you how you're going to be, how you're going to look, how you're going to act. All of that stuff is inside your DNA. These codes are in 2 different base pairs, and they're very, very simple. They are the A, C, G, and T base pairs. A and T pair together, C and G pair together. A stands for adenine, and T for thymine, and C stands for cytosine, and G for guanine. And the way they pair up, it could be A, T, A, C, G, A, T, and on the other side it'd be T, A, whatever I said after that. They would all pair up like that, and each of those strands would have a specific genetic code to make you, you. So your DNA is what makes your eye color, your DNA is what makes your hair color, your DNA is what makes you as tall or as short as you are. Your DNA makes everything about you, and there are tons and tons of this stuff inside of your body. And once again, it's a polymer, and I'm going to show you how to extract this DNA. Not from a human body, but from stuff that we call wheat germ. And we're going to extract it, being very, very safe though. As you can see, I already have on my lab coat, so I don't spill anything on myself. I have on my gloves, so I don't get anything on my hands. So if I rub my eye or something like that, I don't irritate it. And I'm going to go ahead and put on my safety goggles so I don't hurt myself in my eyes at all. All right, are you ready to extract some DNA? Let's do this. Okay, so to extract DNA, it's very, very simple. You're just going to need a very few things. The hardest thing to find is probably going to be the centrifuge tube. This is a 50 millimeter centrifuge tube, but I'm telling you right now, you can get a bunch of these, like this rack over here you see, for like $9.99 on Amazon. They'll send them right to your home. So it's a little bit more difficult to find, but not too hard to find. Next, you're going to need some wheat germ. I'm using Bob's Red Mill, but you can use whatever you want as long as it's high fiber and it hasn't been messed with at all. You just want natural wheat germ, okay? Or wheat bran is fine as well. You're also going to want some soap. For whatever reason, I've always had the most success with this Blue Dawn soap, so it's a really good one to use. You're going to want to get some alcohol. I'm going to be using 70% alcohol. This is why I'm wearing my goggles, because if you get 70% alcohol on your face or hands, you want to protect your eyes. And then you're just going to need various objects like Dixie cups. You're going to need some chopsticks or skewers, and you're going to need some pipettes. Okay, so the first step to doing a wheat germ DNA extraction is you take the top off of your centrifuge tube and you put 5 milliliters of wheat germ into the tube. Okay, now that you have your wheat germ inside, the next thing you're going to do is you're going to fill it up with water to about the 25 milliliter mark. Okay, so once you have it in the 25 milliliter mark, you can shake it up a little bit if you want to get it all mixed in really nice. And then you're going to open it back up and you're going to go ahead and take a skewer and you're going to take it inside and you're going to smash it all up. The reason why you want to do this is your wheat germ cells have a very strong cell wall and a strong cell membrane, and you want to break that apart so that you can get water inside the cell so that the nucleus starts to fatten up a little bit. So after you've smashed up your wheat germ a bit, it's going to have this cloudy kind of look. You're going to have a lot of wheat germ on the bottom. That's okay. If you really want to get it all smashed up, it would take a long, long time. But I'd say smash it up for a good 2-3 minutes. But if you really want to get it good, 10-15 minutes, if you just want to keep smashing it, you'll get a lot of it smashed out. And it's cloudy like this just because it's opened up, like I said. Now the next step you're going to do is you're going to go ahead and take the soap solution over here, and you're going to start to pipette some of that soap solution inside the centrifuge tube. Now, why do we put soap inside? Think about what you use soap for at home. You use it to clean off grease from your dishes or from your pots and pans. Grease is basically congealed fat. So when we put soap solution into the centrifuge tube, the fats, the lipids from the cell will attach to that, so the DNA has a very, very clear path towards the last step I'm going to show you. So next thing you're going to do is put soap inside. After you put soap inside, you're not going to shake it up like this. That'll mess up your experiment. What you're going to do is you're going to take it and just using your wrist, you're going to do something called whorling, which is this. Okay? Now I'm going to tell you right now, you have to whorl for like a good 5 minutes. And you want to get as little amount of soap bubbles as you can. If you have 0 soap bubbles, you've done really good. The reason why you don't want a lot of soap bubbles is the DNA, as it travels up into the last step, which I'll show you in a moment, it'll get trapped into those soap bubbles, and you don't want that. Okay, so your final step is going to be this. You're going to take your 70% isopropyl alcohol, which I also put into a cup over here. You're going to get a new pipette, so you're going to need at least 2 pipettes. I would say you want to get 3 pipettes because one of your pipettes will be used to extract the DNA if you want in a moment. And what you're going to do is you're going to put some pipette-filled alcohol into the cup. You're going to do it maybe 1, 2, 3, 4, 5 times because you want a nice layer of alcohol so that the DNA can travel into it. What you're going to notice is that the alcohol is less dense than the soapy water and the wheat germ solution. It's going to stay on the top. It's not going to mix in with the rest. That allows the DNA a chance to move through the soap solution and into that alcohol layer, and then you'll be able to see it floating around. It's going to look like snot almost, but we'll get to that in a moment. So now you can see that layer of alcohol floating on the top. And if you look, all these little filaments that seem to be rising up from the soap into the alcohol, that is your DNA. And that is what you're going to be able to extract using either another pipette. You can use the alcohol pipette or a different pipette, or you can use a chopstick to pull it out. We'll show you how to do that in one moment. Okay, so right there's the DNA, and there's a couple things you can do. You can either try to grab it like this using your chopstick, And then, yeah, you can get like a nice, you got to kind of whirl it like spaghetti, and you can pull it out. And you'll be able to see it better on your stick than you can on the camera. But the other thing you can do, which is really easy, is you just put a pipette inside, grab it right by the DNA, pull it up, and you can see it's inside the pipette there, that you pulled some out. Okay, so that's how you extract DNA, which once again is a polymer, a very long chain molecule with repeating subunits. And in this case, we're talking about a plant, a strand of DNA. Now, when you get home today, I want you to think about something. We use this wheat germ to extract the DNA. It's still floating in there. You can kind of see it, but not too well right now. But when you're at home, I want you to think about, are there any other items in my house, particularly in my kitchen, probably a food item, that I can also extract DNA from and give it a shot? Now, I will warn you against one thing. Don't try to use meat. Don't try to use like rice and dry goods. Think about other things besides those. Okay? Have fun extracting and hope to see you soon. This is Jason. Bye for now.